Welcome to the afternoon plenary. My name is Michael Roberts. I'm the Deputy Chief Scientist of CASIS. And I have the distinct honor of introducing you to the panel this afternoon, ISS Research to Advance Health on Earth. In an era of increasing human longevity, it is becoming ever more critical to prioritize not only improved treatments for disease, but also approaches for sustaining wellness, to preserve the quality of life for aging adults. Spaceflight-induced physiological changes represent deviations from this state of wellness, with some phenotypes serving as accelerated models of Earth-based conditions, including musculoskeletal disease, immune dysfunction, and delayed wound healing. Research on board the International Space Station National Laboratory seeks to exploit these spaceflight-induced physiological effects to improve human health here on Earth, home, through studies that advance the prevention, detection, and treatment of these diseases. In parallel, spaceflight research and technology efforts also seek to improve the human condition by employing the space environment to advance diagnostic tools and drug discovery, development, and delivery systems. Your panel this afternoon is going to present to you a few of the highlights of research that are directed towards these health-related issues that, were it not for access to the International Space Station National Lab, would not have been possible. Our moderator this afternoon is Dr. Bess Dawson-Hughes, Senior Scientist and Director of the Bone Metabolism Laboratory at Tufts University. Bess?
Thank you, Mike. It is certainly a privilege and a lot of fun to be here and to be a part of this august group. My own background is in endocrinology, and I'm interested in bone and muscle metabolism. Our speakers today are shining examples of success of one of the main objectives of the Center for Advancement of Science in Space. CASIS, as you pretty well know, is directed at technology and health research, health-related research that will pay back to the taxpayers and to people on Earth. So we have three examples here in the presentations that address good health outcomes in terms of the space research. So as a director of CASIS, I am happy to introduce these speakers to you one by one. First, I'd like to say that we will allow several questions after each presentation. And then with time remaining, there will be, at the end, there will be a second shot at having your questions addressed. So without further ado, I would like to introduce our first speaker, Dr. Hughes Fulford, who is a professor at UC San Francisco, and she runs a laboratory that is devoted to lymphocyte activation and lymphocyte performance. She has received many awards during her career, including the 2013 NASA Top ISS Discovery Award. And for that, we are very proud to have you here. She has flown on 2 missions as a payload specialist to the space station, and has sent many other experiments up in addition. She has discovered that microRNAs, their expression is dysregulated in a low Earth orbit environment. And today she will discuss the particulars of some of her findings related to lymphocyte function, miRNAs, and their connections. Thank you for being here.
Thank you for the nice intro. It's good to be here. I'm surprised so many people have shown up, considering it's a beautiful afternoon. I just want to talk about what we've been discovering over the last couple of years, and I'll give you more information. I'm not sure where is the receiver over there? Okay. The reason I study the T cell is because it was one of the first things we found in the Apollo mission. Apollo 7 had 3 astronauts that had a cold during the flight. Apollo 13, the most famous sickness on orbit, Fred Hayes became very, very sick with a Pseudomonas that normally does not affect normal people with normal immune systems. In all, 15 of 29 Apollo astronauts had an infection either during the flight or immediately following the flight. If you think about the T cell, we're talking about the T helper cell. And the T helper cell is like the general in the army that calls the other cells to serve in the war. That includes the B cells to make antibodies. It includes the macrophages being activated and also the killer cells, the killer T cells. There are 3 signals that are required for a full T cell activation. The first is the presentation of the antigen. In orbit, we can either add anti-CD3 or Con-A to do that synthetically. You have to have an accessory cell, and on orbit we use anti-CD28 to interact with the CD28 ligand receptor. And then finally, when you have those 2 signals, they produce an increase in the synthesis of IL-2, and that in turn causes the IL-2R receptor to be activated, and then you have a fully functioning T cell to call the other cells to war. The end result is we have inflammatory factors like TNF, interferon, and other chemokines and cytokines that mediate the T cell. We flew the T cells on SpaceX 3. This is a photograph of our cassettes that we use to activate and fix. We flew with ESA because ESA had a 1G onboard centrifuge inside of the incubator. So if you look inside, you'll see the circular centrifuge that gave us a 1G effect, and then on the outside we had the microgravity cassettes. We then used gene arrays to identify the genes that are important in early activation. The gene array actually, when you process it, has over 2 million dots that give you the information about your controls, information about 8 to 12 of the probes for each gene. And then you put it through bioinformatics. And you see here the differences in the microgravity, the gravity, and those are both activated in the same way. But there's a big difference in the genes that are upregulated between the microgravity and the gravity. And then, of course, we have the non-activated T cell. And it's the usual suspects. We see IL-2, IL-2R alpha, and the black bar is the 1G control, and the gray bar is microgravity. We see an increase in TNF alpha, we see an increase in EGR1, and this is routinely seen on the ground when you activate, but what was interesting is you're not seeing the activation in microgravity. That data resulted in a paper on the NF-kappa beta, real pathways controlling early T cell activation. And the question is, do other animals have the same type of change in their immune system? And we were able to fly mice on STS-131, and they were in the AEM. We had 8 mice in the air and 8 mice on the ground. And again, you see a really big difference between the cells that weren't activated and activated. What was interesting is that at 24 hours, which is the last bar, some of the effects had dropped off. And so we know that the effect of microgravity in the mouse lasts for around 20 hours. Yeah. So whatever changes is still changed when we first bring them back. Conclusions are that the T cell does have a dependence on the immune system. The T cells have a dependence on the response because of gravity, and since now we've seen both people and mice, we know it occurs at least in 2 different animals. The mouse experiment resulted in 2 papers. One, we compared our spaceflight data with the 2 simulated microgravities and defined the differences seen. And the other paper looked at the impairment of the antigen-specific tolerance induction in the mouse. And we have one more paper coming out on the microgravity and miRNA for mice. So many of you are engineers, some of you are biological scientists, but basically this is just showing the course of action. The first thing you see is induction of the gene, the messenger RNA, and the second thing is translation into a protein. But there's another level of control, and that is the microRNAs. And they're small encoding RNAs, are about 20 nucleotides. The first one was reported in the C. elegans, and in 2004 they were found in human. They act as a binary switch, turning protein synthesis off and on. So now we're going to describe the changes that we're seeing in the microRNA. Basically, once you have your microRNA, it gets incorporated into an RNA-induced silencing complex that will then sit on the RNA and act like a block. And so you're not making RNA, it cannot go through the ribosome. And the question was, is it regulated differently in the human system in microgravity? The first miRNA that we looked at was MIR21, and it was just by accident that we saw the difference in it because we were just collecting for messenger RNA and not for total RNA. And what was curious about this is not only did in 1 gravity we turn it on, but even with 0.5 gravity we were able to turn it on. And that brings up the question, do we need artificial gravity in spaceflight? And hopefully we'll be able to do some of that work in the coming years. So we have several genes that are turned on in microgravity, in gravity as opposed to the microgravity, the shorter bar. But what was curious for us is the MIR21 was also being turned on, and we were having trouble understanding why we were turning on MIR21 and its targets. And then we realized that it's probably because it takes a while for the MIR21 to be made and to be mature enough to act. And so we, let's see if we can, there. We now know that it is because even though the miRNA is coming up with the genes, it is causing, once it's made, it blocks some of the primary genes that have to be turned off in order to turn off the immune system, which is important because if your immune system doesn't have internal control, then your cytokines get multiplied to the point that you could have a cytokine storm. So this might be one of the ways that miRNA is controlling the RNA and the protein synthesis of some of the early genes that come up. So anyway, this new mechanism, we published this also recently, talking about the microRNA in spaceflight and T-cell activation. And about 2 decades ago, a little bit longer, we started using biological drugs, and that we started using drugs that affected the biological consequence of T-cell activation. And we have a lot of TNF inhibitors that are used to treat RNA. We have some that are used as a leukemia treatment that are specific for the CD20. And so this was a revolution. I think the same revolution will occur in microRNA as we go on. And what's interesting is all the biological drugs are treating the symptoms at the very big part of the waterfall, and the microRNAs would be looking at the raindrops coming down and controlling it from that level. So basically, I will end with STS-5, the SpaceX-5, and here you see the activated cells, T cells, have a part of the miRNA turned off. And what's curious is those are the miRNAs that are actually controlling the genes that are needed for the early activation. So not only are we inducing activation of the early genes, but we're turning off. So it's break off, gas on. So using the microgravity of International Space Station has helped us find new mechanisms behind miRNA control. And now we have a way to start looking at immune dysfunction in older people or people with rheumatoid arthritis and Crohn's disease. And I'd like to thank a lot of people for a lot of things, NIH, CASES, NASA, for making all this work possible. Thank you.
We have time for a couple of questions. If there is a question, I think someone's coming. Yes.
Hi, C.C. King. I'm from UCSD. Really fantastic and interesting work on microRNAs. I was curious on 2 questions. If you've looked at the dynamics of microRNA expression at various time points, first of all. And then second, I guess I wanted to know if you'd looked at the promoter regions of miR-21 and other microRNAs. We're interested in how miR expression is regulated and whether there's a common transcription factor for that green cluster that you found.
Actually, that's a different paper. But anyway, yes, we found a relationship in the promoter regions. That's the first thing we looked at. And they shared promoter regions that we had in their target genes. So it's made to be turned on when you're turning on your early gene expression, and it's there to turn them back off when it's time to turn them off. So, and time points, we have looked at time points, and you can follow the gene expression over, 48 hours, but we've gone up to 24, and we're seeing a peak at about 4, and then a decay at 24. Thank you for the question.
Hi, Kumar Sharma, UCSD.
Oh, that's better, yes.
Yeah. Could you comment on why you think miR-21 is altered with microgravity, and was that the only microRNA that seems to be the major one regulating the immune system?
The MIR-21 was only found by sheer luck. the second slide with RNA was done specifically looking for miRNA. The reason it's turned on is because it shares promoter regions with some of the immediate early genes that are turned on for activation. The second question, why are the other ones being turned on and off? We're in the process of looking at promoter regions and also looking at mechanisms that MIRs are degraded in a hurry. Thank you.
At this point, I would like to introduce Dr. Rosalind Smith. She received a DPhil in biochemistry at Oxford, did a couple of postdocs in the US, then went to Austria for a few years and worked, and then 25 years ago joined Eli Lilly in Indianapolis. She's in the biotech discovery division and actually leads the regenerative biology unit within that division. She seeks to identify bioproduct candidates for clinical development to enhance tissue repair, and her main focus in recent years has been on musculoskeletal diseases. So she is here today to address the topic, using the ISS platform for drug discovery for muscle wasting diseases on Earth. And if any of you in the room happens to be aging, you will know what we are talking about.
Thanks for that kind introduction and the invitation to come and speak to you today. I'm really excited to be able to tell you about Rodent Research 3, which is a mission that flew on the space station earlier this year that was focused on muscle wasting and drug discovery. I'm going to put my acknowledgements first in case I don't get time at the end, and there are such a host of people to thank for the experiment that went up to the space station. And not everybody's even included on this, but a lot of people were behind this experiment that I haven't got time to thank today. Also, for those of you who aren't familiar with Eli Lilly and Company, just a brief word to say that we're based in Indianapolis, in Indiana. We actually just celebrated our 140th year. And we are a global pharmaceutical company, and we actually, I just, for the purpose of this talk, want to mention that we have expertise in musculoskeletal conditions. So we have a lot of experience in preclinical and clinical measures of bone and muscle. We even have a product for osteoporosis on the market. And so, but our mission really is to make medicines that help people live longer, healthier, more active lives. And so we've been thinking about skeletal muscle atrophy actually for quite a number of years now. And when you come down to it, we actually made a list of all the various conditions and diseases that are associated with skeletal muscle wasting. We came up with well over 100 conditions. Some of these you may be familiar with, certainly muscle diseases like muscular dystrophy and ALS, but you know, certain forms of cancer, pancreatic cancer, there's particularly pronounced muscle wasting often with that disease. Sarcopenia, which is the muscle wasting associated with aging and the elderly. And of course we know about astronauts in space. So, but when we think about skeletal muscle wasting, and I should mention this is therefore not, we're not really talking about cardiac muscle or smooth muscle. I'm talking particularly for this talk skeletal muscle. It's not really the size that matters, it's the function. So when one has muscle wasting, it's really the fact that when the muscle is smaller, you have decreased power, strength, and endurance. And it's that decreased muscle function that actually leads to diminished functional performance. And this is seen in the fact in certain conditions and also with aging that you have the reduced ability to perform activities of daily living. So for example, you You can't climb stairs, you can't go out to collect the mail, you can't put the groceries away, these sorts of things that take some strength to do. And because you are weaker, you also actually are at risk for certain other events. So you have an increased chance of falling, fracturing, also going in the hospital, going into the nursing home. It's actually often a downward spiral that leads to often nursing homes, etc., or to the hospital. And it's generally associated with poor prognosis and outcomes. So unfortunately, even though it represents a large unmet medical need, the only current treatment for muscle atrophy is physical therapy or exercise, and usually that's not sufficient, and particularly with the ill and the elderly, you don't get very good compliance with those sorts of things. So of course we've been asking the question, is it possible to develop a drug to combat muscle atrophy? And I should say we're not the only pharmaceutical company thinking about that, and there are some drugs that are working their way through into clinical development, but unfortunately nothing yet is close to FDA approval. Now, if you're thinking about developing new drugs, you start off needing to get some preclinical evidence for a particular target or a particular drug that you are working on. And so it's important to have animal models of the particular disease that you're interested in. So, of course, then the mouse actually represents a good animal model for muscle atrophy. It's already known that a lot of the mechanisms behind muscle wasting are conserved between mouse and man, so that makes it a useful model. Unfortunately, some of the animal models on Earth for muscle wasting are not always the best. Sometimes you have to just relate it just to one particular limb, or in terms of the way in which the model works, or are particularly related to a particular disease or genetics. So we were so excited at the thought of being able to use the microgravity situation of the space station, where muscles are unloaded and therefore you get wasting, to ask the question, would our new investigational compounds be able to have an effect and overcome the muscle wasting in that environment as a preclinical test of our potential new compound. So this is just, I put together, just from the published literature, and I know there are other experiments also out there that have looked at muscle wasting in rodents in space, but I just pulled from the literature a few of the already existing data on rodent muscle wasting. And you can see that in the space shuttle there were definitely rodents flown previously, short missions ranging from 13 to 17 days, but where it was shown that you get, over that period of time, decreases in body weight and decreases in certain muscles that were looked at. So I'm going to contrast that to our Rodent Research 3 experiment, where we are going for a longer period of time, obviously in the space station, for 6 weeks. We were planning to look at body composition, but we were adding an extra measure, which was actually looking at muscle function. As I said, size and weight of muscle is one thing, but really what's the critical component is actually the function of the muscle. So for Rodent Research 3, our goal was we had a particular investigational compound that we wanted to see if it could increase muscle mass and function in the mice in the microgravity environment, and that we believed would help us in our discovery research to develop new medicines for patients in need with muscle wasting disorders. And of course, there was also the chance to learn other things about how animal cellular functions are affected by longer-term microgravity. We had a few unique features of Rodent Research 3. Obviously, we have a novel investigational compound. The compound that we were testing in mice was an inhibitor of myostatin, which some of you may know, but in a slide in a little while I'll tell you just a little bit more about that. We had a new strain of mice. We had a lot of experience with BALB-C mice, and so that's the strain that we used in this experiment. It was 6 weeks long, as I told you, and we were going to measure muscle function for the first time with a grip strength meter. Because we wanted to do interim measures and measure not only body composition and grip strength halfway through our experiment, not just at the end of the experiment, we actually needed to anesthetize the mice and have them recover to be able to carry on. And so this was the first time that we actually did anesthesia followed by recovery in mice in space. And then also we were going to also look at body composition with the DEXA instrument from TechShot, and I believe this was the second experiment to use that capability. So the experimental design, we had 10 baseline mice, but then we had 20 mice flown in space. 10 of them were control treated, 10 of them were drug treated. And then, of course, we had on the ground equivalent 20 mice that we tried to duplicate everything as possible, that as close to the flight mice, except that obviously they didn't have the microgravity situation. We gave our drug. We started initiation of our drug treatment 1 day prior to launch, and then we also gave it subcutaneously at 2 weeks, 4 weeks, and the experiment was terminated at 6 weeks. Unfortunately, for our particular flight, we couldn't bring the mice back alive, so we had to terminate the experiment at 6 weeks. So that was the design of our experiment. So as we worked towards our launch date, we climatized the mice 4 weeks ahead of time to, the new drinking apparatus and food bars, and also to the fact that they're in the different wire floors, like a bedding. And that all went fine. And then we moved the mice into the Dragon habitat, because since these were going up in the Dragon capsule on SpaceX at 1 day prior to launch. That's me in the photograph, having just helped load the mice into the Dragon habitat. So, as you know, we were going up, the mice were going up on SpaceX 8. We had to wait a long time for that launch to happen, but finally, on April the 8th this year, it was successful, and we got our mice up to the space station. The Dragon docked, and then just over a day after launch, and then the mice were moved into the habitat on the space station. If you can play the video clip, this was some of the activity of the mice about a week after they'd adjusted to life on the space station. You see that one? That's what is called the racetrack behavior. That would be interesting as we come along. And this one there is the pirouetting ballerina behavior. But you can see the mice are very active. They actually all did very well in space, very healthy. We didn't have any issues. And you can see that they were very active. And that may become relevant later on as we get through some of the data here. So 4 weeks into the experiment, we wanted to do interim measures of grip strength, and you can see that here is a video clip where one of the astronauts is going to do some of the grip strength measures. Mice had microchips in them, so that's the astronaut just identifying the particular mouse. And then you can see this grip strength meter. What happens is that the mice hold onto that grid with all 4 feet and hold on, and then you pull the mouse off the grid, and the force that's required to pull it off hanging onto that grid is actually a reliable measure of their actual strength. And we've used that a lot here on Earth to measure muscle function in mice. We had 4 measures taken per mouse per time point, and we averaged the 2 values. And you'll see in a minute when I show the data that it's amazingly tight how the data turned out really well. So then we also looked at body composition using the DEXA machine, which actually allowed us to look at lean mass, fat mass, as well as bone mineral content and density. Because, as I mentioned in the interim measures, we needed to not only anesthetize the mice, but also then recover them. Actually, Bioserve came up with this new apparatus where the mice could be individually housed and warmed while they recovered from the anesthesia. We also measured the grip strength and the body composition at termination at 6 weeks, and then also we did these things too. We collected obviously serums, and we're going to collect all these samples that are coming back to Earth. Hopefully, if SpaceX launches on time on Sunday, we'll get them in late August. And we will be doing a lot of other things, obviously with those samples listed here, but including histology, gene expression, some biomechanics with the bone, etc. So one brief mention that about myostatin. Myostatin is a member of the TGF-beta superfamily, a negative regulator of skeletal muscle mass. So when you inhibit its function, you actually get an increase in muscle mass and function. We know that from our experiments on Earth. And if you look on the graphs on the right-hand side, on the top graph, you'll see that when we dose with a myostatin inhibitor, whether it's 1 dose or 2 doses, we see significant increases in body weight already by 7 days and 2 weeks after we start treatment. And we can see that in terms of increase in muscle mass as well as grip strength, which is the bottom graph. So the question is, what data did we get from Rodent Research 3? And so I'm excited to show you some of our actual data from this experiment. If you focus on this graph, and I'm going to point to it so it's clearer. If you look at the line here is the grip strength of our, back one slide please. This is the grip strength of the ground control. Here in the blue triangles is the grip strength of our ground controls treated with our compound. So you can see, as expected, we got an increase in grip strength function when we treated with our compound. Here is the grip strength at 4 weeks and 6 weeks of our mice that were in flight. Back one, please. And in green is the flight mice treated with our compound. We got significant effects in terms of grip strength, muscle function in our flight mice. So we were able to conclude that inhibiting myostatin can affect increased muscle function under the microgravity conditions of space station. Next slide is the data with, if you could go to the next slide, is the lean mass data from the DEXA machine. Again, this lower line is the ground controls. The blue triangles show the increase in lean mass at 4 weeks and 6 weeks when we had treated with our compound. So we got a significant increase, change from baseline, in lean mass with our compound. Here is the lean mass that we got with our flight animals, and again, the green squares are the treated animals. Again, significant increase at both time points with our treatment of our compound. However, the astute among you may notice that our flight animals were, if anything, slightly higher in terms of the increase in lean mass compared to the ground controls. That was not what we expected. We expected to have a decrease in lean mass since we expected to see muscle wasting. We did not see that. So the question is, well, what was going on here? If you go to the next slide, we asked initially, well, was that because the ground controls were performing poorly? It wasn't that why was there a difference? But we actually had done an experiment at Lilly on using the same mice, exactly the same protocol, where we looked at and treated with the same compound in the same way. And if you look at these are the Lilly experiment, this is the ground control experiment that was done in Cape Canaveral. You can see that we got the same effect on our compound, the same growth of the mice, this is in terms of body weight. So we don't believe that the ground controls were performing poorly. So what is the explanation? Could wasting occurred early and then at this later time point where they've recovered or overcompensated. We still ask, could somehow, I don't know, fluid distribution affect our DEXA measures? Or could it be that extra amount of all that wonderful exercise and behavior that we saw in those mice, could that be having an effect in terms of just extra exercise of those mice that compensate us? We don't know that. We'll see when, obviously we're waiting for the samples to validate the DEXA measures with actual muscle weights, etc. And when we get the samples back, and then we'll really know the answer. So this is my last slide, just to say that obviously we've, in summary, been able to show that our compound, our myostatin inhibitor, was to be able to show significant effects on not only muscle function, muscle size under the conditions of microgravity in this experiment that we performed on mice in space, but the unexpected finding was that we didn't see the decrease in muscle mass at those time points that we looked at. So we need to continue to analyze the samples and the data when we get them back, and we'll be anxiously looking and asking a lot more questions to see what is going on there. And of course, the data we'll be able to use to understand further the effects of microgravity on muscle and to aid our main goal at Lilly, which is to find new drugs to combat muscle wasting diseases. And hopefully, maybe next year, we'll have more data and be able to come back and tell you more. Thank you very much.
Are there any questions? I might just make a comment while someone is coming to ask a question. And we've done a lot of work with DEXA technology over the years, and that technology does not distinguish between lean mass and water mass. And so I think that's probably a contributor to your observation.
Well, that's why I asked that question, but I know the tech shop folks are in the audience, and they may be able to comment if they want to make a comment in regards to that, because that gets into some technology which is beyond me, but happy if they wanted to make a comment. But I agree, that's why we put it down there as one possibility.
Yeah, maybe.
Hi, Jim
Hello.
Kirkpatrick from AAS. Following the whole progression of these mice in space, I kind of wondered how it was going to end, and when you said termination, then I knew. I don't want to know how mice are terminated by the astronauts, but is there any value to ever bringing samples like this or mice that have you administered these protocols to bring them back alive and have them further tested or in that environment? Or I know probably it's helpful to have the actual samples and look into the muscle tissues and everything else.
I think that at the time that we designed and were working on our experiment, the capability wasn't there to bring live mice back. But if I'm correct, I believe that on Rodent Research 5, they will be bringing mice back. Is that right? And so I think that now is something that is going to be possible. So I think I was in the technical session this afternoon listening to some of the designs of those experiments, and they will actually be keeping some up there, bringing some back down, etc., and comparing. So I think that's now possible. It just wasn't possible at the time that we designed our experiment.
Okay, thank you.
I just have a couple questions. I was interested in the way that the mice were moving. I've never seen an astronaut do it like that. But I was wondering, you mentioned about the muscle, the activity. It was a higher activity on the space station, or did you have cameras in the box on Earth, or did you compare their activity?
That's a good question, because again, I don't know that, and Lewis, you're in the audience, and you can answer that question for me. Lewis Studiak from... Yeah, and in fact, actually, again, in one of the technical sessions on rodent research, one that I saw this afternoon, they actually looked at activity at a certain point in time between the controls and the flight animals, and actually saw a significant increase in activity in the flight animals compared to the ground animals. I think I saw.
Okay.
So
But
that was
did
rodent
they-
research. Sorry.
I just, so when they were sleeping, did they continue to grip the cage to feel comfortable, or did they ever let go and float?
I don't know, since that's probably in the dark cycle. I don't even know that we know that, so I'm sorry. Don't know the answer to that offhand.
Take that offline. Okay,
Yeah,
thank you.
take that offline. One quick question.
One more quick question, or maybe two.
Thank you for your talk. I have a question about the fact that you are an original manufacturer of the medical or drug systems. Studying in the space with this very small sample, how do you deal with the FDA compared to, this is a preclinical trial. I understand that you can have a smaller sample, but considering that what we can do on Earth, you have a very smaller samples. How do you deal with FDA with that? Thank you.
Well, it is not going to be the only experiment that we would use as we collected preclinical data to support moving a drug into clinical development and beyond. So it is an important piece, it's exciting data, but it's only one piece of the whole data package that you would have to produce.
Alan Hargens, UC San Diego. I was interested in your data. Are you going to be able to measure or look at the soleus and anti-gravity muscle specifically?
Yes, we plan to do that, of course, not just gastroc and quad, but we've got a list of muscles that we will be taking out and looking at. That's one of the muscles that we routinely look at in our experiments, our regular experiments here on Earth, soleus as well as gastroc and the quadriceps as well as other muscle types.
Yeah.
So we will be looking at that for certain. An important one, I think.
Good. And I think your experimental design is excellent. I think it's better to harvest the animals while they're in space as opposed to letting them come back to Earth and readapting to Earth, which usually takes hours, maybe 12 hours before those animals can be dissected.
Yeah, that's a good point. Thank you.
Thank you, and that concludes this segment of the program, and you were all, I think Mike may have a comment, a closing comment. Thank you for your participation in this.
Yeah, please thank the panel one more time. That was a great presentation today. So thank you for your attention. I'm not going to hold you up long. This session started this morning with a view of new capabilities provided by launch service providers, and you heard about the innovations that are happening there to drive price points down. And in the middle of the day, you heard some very innovative thinking about new environments, new asteroids to mine, new thought processes involved where the advancements are not only very small, but are actually very grand in scale. Several orders of magnitude are possible in these new disruptive innovations. And I think the panel closing today provides to you that there are opportunities across a variety of biomedical needs that can be addressed here, that are dire needs here on Earth, that the space environment can offer new avenues, new approaches to finding new therapies, new cures moving forward. And most importantly, the thing I think tying all those together is that most of us in this room, I think, are enthusiastic about the possibilities of utilization of space as a research and technology environment. And we probably got that bug somewhere along the way when we were younger. And I wanted to remind everyone that tomorrow is officially STEM Day here at the ISS R&D conference. So you heard a lot of the efforts that are afoot to combat the effects of aging. Tomorrow, we're going to bring the age level down quite a bit. There will be some students here who will also be helping us be more motivated and more engaged. And I wanted to especially call out in the morning, beginning at 8 a.m., there's an opportunity to, in this room, hear the winner of next year's Genes in Space program. So there will be an opportunity for that at 8 a.m. At 8:45 a.m., there's the amateur radio on the ISS. So there will be an opportunity to hear several schools who are linked together, and will be able to have an amateur radio uplink to Jeff Williams on the International Space Station in real time. And that's among several other activities that will be going on tomorrow. So please have a good time with the reception tonight and the posters. Have a good time in San Diego tonight if you go out, but be back here at 8 a.m. in the morning ready to go. Thanks, everyone.